Okay, so let's get started. It's already 5 p.m. We'll join, share my screen. Yeah, okay. Okay, so welcome to week 7 for NGS data analysis and applications. So, in last week, we discussed about different type of variant calling. We discussed how the VC format looks like. We discussed different types of algorithms and different types of logic used to call variants and how to store those variants and use them downstream. So we talked majorly about SNVs in the last class. We touched upon the concept of SVs, but in this class we'll talk about mostly about SVs and how you can like identify SVs with different types of techniques. What are the different types of callers that are used to identify the structural variations? And after that, we'll talk a little bit about RNA sequencing and the different types of caveats related to RNA sequencing and how like different pipelines for RNA sequencing data analysis will be different from genomic sequencing data analysis. So let's get started. Okay, so first comes the concept of structural variants. So we talked about SNVs in the last class. So SNVs are single nucleotide changes in the genome, which may or may not have an impact on the overall expression of the genome in terms of gene expression, in terms of other epigenetic modifications or any other things. They can be benign or they can be protein coding changes as well. So if inside the gene, if a single nucleotide variation occurs in a place where it's the exon part of the gene where it is like translated downstream to a chain of amino acid and protein. So in those cases, what will happen is if there are single nucleotide changes, uh, the translated amino acid from the mRNA, from basically from the genome to the mRNA to the translated amino acid. So the single nucleotide changes can like disrupt this amino acid synthesis, and in also some cases it can just change the amino acid that was originally planned to be in that position. One such good example is sickle cell anemia. Uh, so one nucleotide change leading to a basically a different amino acid in place of another one that causes the disease. So structural variants, on the other hand, are like big variations. These are like not single nucleotide or like 20 nucleotide. We generally define structural variants, which are like variations which are greater than 50 nucleotides in length. So I'll just check quickly if anybody has joined or something. Nope. Okay. Yeah, so structural variants are variants in the genome which are generally 50, more than 50 base pairs long. That's how we define it. Uh, yeah, just give me a second. Yeah, and they can be further subdivided into major types of structural variants, including deletions, insertions, uh, duplications, inversions, and translocations. So there can be many types of structural variations. These are like the overview uh, of the 5 categories that are majorly present. But generally, how we define structural variations is quite uh, just a second. Just give me a second. My laptop is charging. Yeah. Okay. Somebody is trying to join. Okay, so the structural variations that we define are mostly like greater than 50 base pairs in length, and they can be like different types of structural variations. So in one category are the category where we call copy number variations, where basically the number of copies of that particular DNA stretch increases. Like that we what we see in duplications, and also it can happen that the number of copies decreases as well as we see in deletion. So these are like copy number variations, and also there can be another set of structural variations which are non-copy number variations. So for example, an inversion. So a stretch of the DNA has been inverted, like the sequence has been just been reversed. In that case, the copy number does not change, the amount of DNA does not change, but obviously the genome changes, and if those are within like the region is within a protein coding region or an exon of a particular gene, it'll affect the protein formation downstream when translation will occur also. So here are depicted like five major types of structural variations. This is the deletion. So like the upper strand is a reference, and the strand on the below is the sample, and we always compare the sample to the reference to find out what kind of variations are there. So this in the case of deletion, so these dp and sr and rd, these are three important parameters by which we kind of discover structural variation. So rd is known as read depth. So recall from the first class, so read the depth or the coverage of a genome is basically how many reads are supporting a particular position in the genome. So if you kind of think about it, so in copy number variations, if there has been a duplication in the genome, so the number of reads that are going to map to a particular that particular area in the reference genome will kind of increase if there is a duplication. If there is a deletion, the number will decrease. So mostly copy number variants are like discovered using read depth techniques, read depth not techniques, I should say, read depth measurements. Also, there can be other forms of evidences for a structural variation inside a genome. So one of them is a split read. So like if you're trying to say align, say for example, this particular uh, read to the reference genome, what you'll find is a part of the the read is aligning to the left of the structural variant. Sorry. Yeah, a part of the read is aligning to the left of the structural variant, and another part is aligning to the right of the structural variant. So this part will be missing. So a same read will be aligned in two different portions of the genome. So these kind of evidences we accumulate using different types of structural variant callers, and we kind of predict what kind of a structural variant is present in that part of the genome. So we'll talk about this evidences later down in today's class, but just to get an overview. So mainly these are the types of evidences that we use. We also use paired read evidences. So if you are using a sequence, Illumina sequencing machine, and you have generated reads, so from read pairs and orientation of the read pairs and the distance between the read pairs can also tell you if there is a structural variant present in that particular part of the genome or not. So moving on. So in deletion, you'll have like a part of the genome that is missing from your sample but is present in the reference. Insertion, it's opposite. So it'll be present uh, in your sample, but it will not be present in that portion of the genome in the reference sample reference genome. In duplication, just the number of reads will increase, mapping to that particular region in the in the genome, indicating that the copy number has increased. In case of inversion, it's quite interesting, where we use kind of this. These are like paired reads. These two opposing reads that are shown here. So these kind of reads are generated from Illumina sequences, as we have discussed earlier as well. So these kind of evidences are looked for in cases of inversions, and in translocation is a little bit more complicated than these four classes of structural variant. In translocation, a part of the genome is cut from one place in the genome. That might be another chromosome, that might be in the same chromosome, but a part of that genome is cut and inserted in that position. So it's hard to kind of unearth these translocations. But one good way would be to use split read evidence. So in split read, one part of the read will align to your to the that area of the reference genome, like to the right or the left of the structural variant, and the other read will align to another part in the chromosome. So these kind of evidences we accumulate, and we claim that a particular structural variant is there. Complex variant is another type of structural variant. Like it's not, you cannot define it as another type. per se, but like it's a combination of these different structural variant in one particular position in the genome. So uh, clusters of breakpoints in genomic regions which cannot be classified are considered as complex SVs because we do not know. So in that portion of the SV, there might be a deletion, also an inversion, also some sort of other SV or anything else. So those which cannot be directly classified into these like four major categories, we call them complex SVs, and they are likely to result from either progressive rearrangement or a major genomic disturbance. So, for example, as I was saying, one portion of the genome has been cut from that place and pasted here. So, these type of rearrangement, these are like quite major genomic rearrangements, which can lead up to diseases or different types of phenotypes that can be seen in individuals. So, uh, like these are complex SVs, and these SVs, these are basically the red blocks are denoting the SVs here. So these SVs are characterized by breakpoints and reads aligned to flanking reference sequences. So these are your flanking reference sequences. In this case here, in this case here. So the reads generally align to the flanking reference sequence, and your structural variant is kind of, and from that information you can gather key like where your structural variant is, how long it is, and what kind of a structural variant is there. And the reads directly below the DNA strand represent the distance and orientation at which they are generated during sequencing. If the reads align differently than expected to the reference strand, this is an indicative of an SV. As I was saying, if one read aligns to one part of the genome, another read, like the same paired read aligns to a different part of the genome, you can say like maybe there has been a translocation that has taken place. And changes in read depth or coverage indicate mostly larger duplications or deletions and are useful for detecting copy number variants. As I was saying, like in duplications, the number of reads mapping to that particular portion of the genome will dramatically increase, or if there is a deletion, the amount of reads mapping to that particular portion of the genome will dramatically decrease. So you can visualize this in the IGV browser. If you have a BAM file, you just upload the BAM file and you can visualize the portion of the genome where the structural variant is and what are the reads, how many reads are mapping to that particular position. Is there is an increase of the number of reads or a decrease? You can like visualize using different kind of softwares. IGV is one of them. And discordant pairs, which basically are paired reads or normal reads that are not uh, correctly oriented while aligning to the reference genome, meaning after alignment, you can understand that the read is not correctly correctly oriented at least. Maybe like a part of it is aligning here or it is aligning in an opposite direction, stuff like that. So those are known as discordant read pairs. Uh, Aligned to the reference at different relative distance and or orientation than expected. DPs are best suited for detecting large SVs such as intrachromosomal translocations or inversions. Split reads span breakpoints and can only be partially aligned. Split read can detect small variants with base pair resolution, especially those smaller than the size of the read. So here comes the an interesting point. Key the split reads are they are like very good indicators of structural variants. So if you have a read long enough to span the structural variant. Now, normally in Illumina we have in a whole read we have the read pairs like 150 base pair, 150 base pair, and there's an insert size inside it. So around if I assume like around 300 base pairs, so if the SV is around 300 base pairs, or in the if you are using a single read sequencing technique, if the read is above if the SV is above 150 base pairs, then one split read from the evidence of one particular type of split read, you won't be able to claim a structural variant there, because the structural variant itself is around say 500 base pairs, but your read length is 150 base pairs. So your read would not be aligned in a split manner. So it will not span the whole structural variant. So here comes the importance of long read sequencing. So remember in the first classes we talked about how long read sequencing can enhance your power to detect structural variants. So here comes one such example why if you have a read long enough, if you have 1 kb read, and if that read is getting aligned in a split manner in your reference, you can claim SVs up to say 1,000 base pair reads length by with the help of one single split read evidence. So, but these like things, these big SVs cannot be well classified in terms of in when you are using short read sequencing techniques like Illumina. So we only have around 150 base pairs. So anything more than that, we need further evidence other than split reads. We need some sort of say read depth evidences, or like since they are paired reads, distance between pairs and those kind of stuff. So it becomes easier in long read sequencing to identify SVs, which is a little bit more tedious in case of short read sequences like Illumina. Okay, so what we talked about in the first slide in the four different types of structural variants. Uh, just a second. Nobody else has joined. Okay, the things that we talked about in the first slide are like the major types of structural variants, duplication, deletion, insertion, inversion, those things. So those are what are termed as simple structural variants. So there's like one type of structural variant in that particular area of the genome, but there can be other types of like complex structural variants. How we call it is basically multiple types of structural variants in one portion of the genome. Say for example, take this example. So you have a reference here. So these are assuming there are two different portions of the genome. You have a so this whole read, if you define it as A, and this whole read you are defining as B. Now when you are in your genome, in the sample which you have sequenced, you find that the A is not there, only the B has been duplicated. So there are two events that are going on here. So A has been deleted and B has been duplicated. So this is a deletion plus duplication. So which is a complex variant. In a similar manner, in this case as well, you have a and a B. Your A has been deleted and B's orientation has been reversed. So this is a deletion plus the inversion structural variant. So these are the types of structural variants what in which we call as complex structural variants. So these are not one single type. So there are multiple types of things that are going on in that portion of the genome, and it's very hard to classify them into one single category. So and these are the arrows that you see on top of these pictures are different types of evidences that we have. So this blue, different types of blue arrows are your inverse read pairs. These red arrows are your deletion read pairs, and this green arrows are your duplication read pairs. So using different types of evidences, you can find out what exactly is going on in that particular portion of the genome. But saying having said that, there are limitations. As I was saying, if you have long read data, it's much more easy. uh, it's much more easier to find the structural variations, because in most cases the uh, uh, the the read length only will span over the whole structural variant, which is not the case for basically other types of like short read sequences. If you, in most cases, the structural variants that we find are around the like 300 500 base pair range. So if that happens, like it's very hard to uncover that structural variant with a 150 base pair read Illumina sequencing technology. So yeah. Okay, so this slide kind of uh, in, encompasses all the different types of methods that we use for um, detecting structural variants, and on the right, this table is telling you basically what are the different types of algorithms or tools that we softwares that we have. Most of them are like what to say open source, so you can find these on GitHub, and the papers associated with it are given in the reference column. So as you see, there are different, there are like many structural variant callers, out of which like there are some uh, which perform very good, some of them which are not so good in certain situations. So you actually which software you are going to use depends on what kind of question or what kind of structural variant, or like what kind of sequencer you have used. It depends a lot on that. So you have these like a long list of these structural variant callers. Some of them work on WGS samples, which is like basically the whole genome, and some only work on exome sequencing, like exome sequences, like exome CNV is one such example. So And on the method is basically the different type of evidences that it uses to uncover structural variants. So for example, if you take callers like tele or lumpy, these are like quite widely used callers, so they use both read pair and split read evidences. And now there are other methods, like it's not included in this particular slide. Here, so this is called a manta. This is also a structural variant caller that we kind of regularly use for WGS structural variant calling. And this kind of, if you go into the paper and check out what basically they are doing, so you'll find that they are using kind of all possible evidences that are there. Uh, yeah. So if you read this, you'll find that like they're using all possible kind of evidences that are there. So in this case, sorry. mm, Yeah. So in this case, there are different structural variant callers which are using either one or a combination of two, three different types of methods to detect structural variants. So let's look into the methods a little bit more. So first comes the read pair methods. These are based on the evaluation and the span and orientation of the paired end reads. So this technique is mostly used for paired end data. So if you are using a Illumina sequencer, if you have paired end reads that you have generated, and from that you want to find structural variants, read pair technique is a good way to do so. And discordant pairs, as I was saying, like the two uh, what how do we define discordant pairs are basically two reads whose either the distance between the two reads have been altered somehow compared to the sequencing time, or the orientation of the reads have been altered somehow, or basically the relative positioning of these two reads is what tells you if there is a particular structural variant there or not. If it is okay, if the relative positioning is like exactly the same as uh, when you had sequenced, then that means that there is not a structural variant. If the say the distance between the reads increase, that means there has been a deletion. If the distance between the reads decrease, that means there has been a insertion. So discordant pairs in which the mapping span on or the orientation of the read pairs are inconsistent with the expected insert size are collected. Read pairs mapping are far too far apart are associated to deletions, while those found closer than expected are indicative of insertions. Furthermore, orientation inconsistencies can represent inversions and a specific class of tandem duplications as well. So as I was saying, the orientations is also important. So they should be like opposing read pairs. If the read pairs are like kind of point like the 3 prime to 5 prime are in the same direction for both the read pairs, which means there has been some sort of inversion of the sequences. Uh, so this goes for read pair methods. The other type of method that are used are read depth or read count. It assumes that the random uh, Poisson or a modified Poisson distribution in mapping length, and it investigates the divergence from this distribution to highlight duplications and deletions. So basically, uh, you have a coverage, you have a certain coverage of the genome on a certain depth of coverage of the genome. So basically, say for example 30, if you have, so on an average a particular base is sequenced 30 times, what it means by coverage. So that means there are at least 30 reads that are kind of providing evidence for that particular base inside the genome. Now, what happens is if you look through the whole genome, the 30 reads are not consistent everywhere. Somewhere it is like 35, somewhere it is like 20, somewhere it is like 50, somewhere it is less again. Like it goes on like this. So how we define depth of coverage is basically the average of all these numbers. So it is already assumed that like the coverage will not be consistent across the genome at every point in the genome. There will be some, like in some cases it will be more, by some cases it will be less. Not because of structural variants, but because of the sequencing. No, I'll not say issues, but because of the sequencing protocol, how how it goes on. So it assumes. So the software callers, the structural variant softwares, they assume a random or a Poisson distribution in terms of the number of mapped in mapping depth and in basically if you see the distribution of the read depth across the genome, what you'll find it's kind of a Poisson's distribution. Now, when it diverges from that, it highlights different types of structural variants. Say, for example, your read depth is 20, 25, or your average coverage is 25. Now, at one point, you see the coverage has gone up to 100. Now, like 25 and like 50, also, I won't be too concerned. But if it goes up to 100, that means something is wrong there, right? So, this is a major diversion from the distribution that you generally expect. So if there is a major divergence from the distribution, they highlight basically duplications and deletions in terms of copy numbers. Like if the copy number has increased, the depth of coverage will increase automatically. If the copy number has decreased, the depth of coverage will decrease, like that. And sequencing of these duplicated and amplified regions result in higher depth, while deleting the deleted regions show reduced depth when compared to the normal. Okay, so we studied about read pair, we studied about read depth or read count. Then next, uh, and the most important method that is used for detecting SVs is the split rate methods. So they allow for detection of SV with a single base pair resolution, because it's like it's the best kind of evidence you can get if you kind of think about it. So if you have like one read that is kind of telling you like a portion of the read is missing inside the reference genome. That's a very good evidence for an insertion. But as I was saying, for techniques like read depth, it's not actually consistent across the whole genome. So you need to have a statistical idea about what is a significant deviation from the distribution and what is not, and what is actually a sequencing error. But in case of split reads, the evidence is very clear. Okay, like if a single read is mapping to two different positions in the genome, that means there is a very high chance that there has been an insertion or a deletion or different kind of SVs that has taken place. Now, this allows for detection of SVs with a single break pair resolution. The presence of an SV breakpoint is investigated on the basis of a split read sequence read signature breaking the alignment to the reference. So a gap in the read is a marker of deletion, while stretches in the reference reflect insertions. Theoretically, all forms of structural variations could be investigated by de novo assembly methods as well. So, like these are three different major techniques that are used. The Manta variant caller that I showed you, basically this one. Yeah, this also encompasses the de novo assembly as well. It tries to assemble the sequence in a de novo manner, and by that method also it finds different types of structural variants. Okay. Uh, Now, what is de novo assembly? De novo assembly refers to merging and ordering short fragments to reassemble the original sequence from which the short sequences were sampled. NGS data intrinsic characteristics such as read length limit the uses of de novo assembly approaches for variant investigation. So it's a more complicated computational approach than read pair, read depth, or split read method. But actually, this is also a good evidence to have when you are discovering structural variants. So what happens is, in most of the cases, structural variant callers uses multiple of these evidences, and they accumulate multiple of these as evidences for a single particular structural variant, and then they kind of call that structural variant and like assign a confidence score or a Fred call score with associated with that particular variant. So uh, like, these are the different types of structural variants, and how we determine these structural variants with the help of different type of evidences. Moreover, a specific class of SVs called as mobile element insertions can be investigated exploiting the discordant and the clipped read information as well. Okay. So this slide basically shows you what happens, what is exactly the difference in terms of detecting structural variants comparing a short read mapping with a long read mapping. So this the upper mm, uh, basically the sequences above is the sample genome, and one in the below is the reference genome. Now if you notice, uh, like in case of, mostly in case of deletions and insertions as well. So these two types of these two types of SVs are mostly different, like in terms of evidence gathering, is mostly different in terms of short read and long read mapping. So if you have a long read like this, and if this is a sample reference, a sample genome, and this is a reference genome, just this has been inverted just to uh, like, oh no, sorry, okay, this is the sample genome, this is the reference genome, okay, sorry. So up on top is the sample genome, on bottom is the reference genome. So in this case, this is your sample genome, this is the read that spans it, right? But if when you're trying to align that particular read to the reference genome, what you'll find is a middle portion of this read, so this portion of the read is not aligning, the end portion is aligning, for the like the start position is aligning, but the middle portion is not aligning. So this is indicative of an insertion in the sample. This sequence is not present in the reference genome in that position. So that means that sequence has been inserted in the sample at that particular position. But one thing to keep in mind is basically this split read for detecting insertion with a single split read. What has to happen is basically this split read has to cover the whole like stretch across the whole structural variant. So if this is a big enough uh, insertion, what will happen is, for in case of smaller basically your short read data, the reads would not span this whole structural variant. In case of long read, it the since the read is bigger in size, say for example a 1 KB read, it will completely encompass the whole structural variant in that particular position, if the variant is obviously lesser than the size of the read. But in most cases, if you want to uncovering like, variance in the range of 700 800 base pair range, it is kind of uh, advised to use long read data, because it's like you won't get 700 800 base pair range in short read Illumina sequencing. So you have to go for long read. But if you do not want, do not have the cost or kind of the infrastructure for long read, you do short read, but you cannot only kind of claim a structural variant present just by the evidence of a particular split read, because that is not possible. So you have to introduce other evidences as well, which includes read pair or read depth evidences. So this slide kind of encompasses, I just gave you this single example, but this slide kind of encompasses the whole differences that are captured in terms of short read and long read data in the context of structural variant. And if you do assembly, so these are the different types of like, so on your y-axis is your sample, on your x-axis is your reference. So in terms of deletion, this basically this portion is not there, and like you'll find a signature somewhat like this. In case of duplication, you'll find these regions of the genome which has been duplicated in the sample. In case of inversion, it's quite evident from this. And in case of translocation, what will happen is one is present in this portion of the genome, another is present in this portion of the genome. So these are different types of evidences and different types of sequencing technologies, how they vary in terms of structural variant detection. So this shows a comparison between de novo assembly, short read, and long read mapping approaches to identify structural variants. For de novo assembly approaches, the relative positions of the segments in the dot plot indicate the type and the size of the SV. For short read mapping approaches, paired end is very important for short read, because the split read won't have that length. So what you'll do is you'll use a proxy, which is basically the the distance between the two pairs of reads. Okay, so for short read based mapping approaches, paired end reads and split reads are typically used to decipher the type, size, and the location of the SV. In addition, the coverage can be used to improve the deletion Yeah, detection of deletions and duplications. Long read based mapping approaches typically leverage alignment patterns of the long reads to different to detect different types of SVs because your read is big enough. Okay. Okay. So these are this brings the end to the structural variant calling part of it. So I'll suggest you can use any of these softwares that are available. Yeah, yeah, you can use any of these softwares that are freely available on a smaller set of data. But yeah, like you can try using different types of these softwares and kind of compare the results, which is giving what like i- in some cases what you'll find is a particular structural variant color is giving you uh, deletions better. In some cases you'll find insertions better. So there are different types of benchmarks already done on data sets. If you want, just go to the GIB website, and this is the FTP of the GIB website. So here you can find, uh, uh, sorry, yeah, here you can find different type of like kind of benchmarks that are available. The samples are also available, GIB samples as well. So you can just take a look in this, and basically I'll just take you to the GIB website. So this is known as genome in a bottle. So these people provide benchmarks. So you can just look into the different benchmarks, and like you can take the same sample, run different variant colors, and kind of compare with the benchmark, which is giving a better one. Okay. So nobody's here, but still I'll send it here if somebody joins or somebody rejoins. Okay. Now we come to the part of transcriptomic analysis, or basically RNA sequencing. Now there there is one thing that is fundamentally different with RNA sequencing, like compared with genome sequencing or DNA sequencing, is basically for RNA sequencing also. In most cases, what we do is we convert the RNA back to the DNA and sequence the DNA, because like all our pipeline, all our like sequencing techniques and everything are optimized for DNA sequencing. So like in most cases what you do is you convert the RNA, use a reverse transcriptase enzyme, and you bring back the RNA toward complementary DNA, and you do the sequencing as done before. So basically this is the workflow that kind of follows. So you extract the RNA, you purify the RNA using mRNA purification techniques. Then you fragment the mRNA and use reverse transcriptase enzyme, which basically converts the RNA to a DNA, complementary DNA. So this converts the reverse transcription converts the RNA to the DNA to DS double-stranded and the complementary DNA. So one thing to keep in mind: RNA is single-stranded, DNA is double-stranded. So basically, uh, to sequence RNA, you need to convert it to a double-stranded DNA and then sequence it, right? So, next steps are the same as genome sequencing as well. So you have to use adapter ligation, you do amplification of the reads, and then you put it through a sequencing machine, which gives you a raw data, and then you do downstream analysis of the raw data. Okay. Now that's the kind of the NGS technique for using RNA sequencing, but the the older techniques include microarray technologies for gene expression analysis. So it's a similar kind of a situation that we saw with DNA microarrays as well. But microarrays kind of are more popular in terms of RNA sequencing because it's more easier to do and gives you a accurate, better kind of result compared to a DNA microarray. Because in the DNA microarray, what will happen is you are kind of trying to get the variants or basically sequence that particular portion of the DNA, but that DNA, like since it's a much bigger uh, stretch of nucleotide, you need to have more markers inside your microarray plate to kind of get a better representation of the DNA sequence. But in case of RNA, since you're only sequencing a very targeted portion of the genome, if you think in terms of that, so it's basically actually a very targeted sequencing of the genome, because the you're getting the RNA, you're reverse transcribing it to the DNA, and then you're sequencing that particular portion of the DNA, which is, if you think in the bigger context, in the DNA, that portion will be just the exons of that particular gene or the particular mRNA. So you are not actually looking into introns, you are not actually looking into intergenic regions, you're just looking into the exons of the DNA. So it's a much smaller subset and a much smaller subset of markers that you have to put on a chip. So basically RNA uh, analysis, or basically gene expression analysis with microarray, is much more popular than the kind of genome sequencing, DNA sequencing with microarray. So the workflow kind of remains same. Uh, so you have the the samples, you extract the total RNA, you cut off the poly A tails, and you kind of use reverse transcriptase to get the complementary double-stranded DNA. And then you use biotin. In this case, biotin is used as to label the transcripts. So you use biotin to label the transcript, and this kind of goes and helps you bind with the sorry this. Then you have to chop it off and incubate in the gene chip and put it on your basically microarray. So in the array, the DNA will hybridize with the complementary sequence that is already present inside the array. So one thing to keep in mind, that when you are designing experiments with microarray, it's a very targeted approach. You need to have an idea what you are trying to get. So for example, if you're trying to sequence a particular if you're trying to get the gene expression of a particular mRNA, you need to know the sequence of the mRNA previous hand, so that you can have those probes inside the chip, and with the help of those probes, the DNA can come and bind here. So yeah, it's the same issues that we faced during DNA microarray analysis, but it's in a lower scale, because you are mm, only trying to get a, a very small subset of the genome. So exons are kind of, if you even take all of the exons, they'll kind of make 10%, less than 10% of the genome. So it's actually a very small subset. So it's easier to do, and so it's basically a very popular method to do gene expression analysis. Okay. Yeah. So you put the you put the DNA inside this chip after converting it from the RNA using reverse transcriptase, and then you kind of amplify the signal and like take uh, pictures from above. Basically, if it comes and hybridizes, there will be some sort of luminescent signal which the system captures, and after processing, you can get an idea about how many meaning how many counts of transcripts have been there, what is the expression of this particular gene. Okay. Okay, so there are some issues when you come to NGS sequencing uh, of RNA, basically mRNA sequencing. There are some issues that needs to be understood. Okay, so this portion, it's basically the preparation for the different types of. So we will be comparing short read and long read uh, sequencing in terms of mRNA sequencing for this slide. So for both short read and long read, this portion is common. So the short read and the long read cDNA methods share many of the same steps of the protocol, but all methods require an adapter ligation step, and are all affected by the same as by sample quality and computational issues upstream and downstream of the library preparation. So this part is the library preparation. Next comes the actual sequencing part. So This is an overview of the three sequencing technology. Basically, like this Illumina is short read, PacBio and Oxford Nanopore are long read, but the technologies are different. So PacBio uses hi-fi reads, and Oxford Nanopore uses like changes in ion flow or changes in current flow through a pore to tell you the sequence of the particular read. Now, if you compare the short read with the long read direct RNA sequencing methods, if you compare this short read and long read direct RNA sequencing methods, over 90% of human genes, which is given by this gene N in this case, are alternatively spliced to form two or more distinct and expressed isoforms. So, uh, in, if you remember, so prokaryotes and eukaryotes are different in terms of their the processing of mRNA. So mRNA is kind of processed differently in eukaryotes and prokaryotes. Eukaryotes have this mechanism called splicing, which is kind of the whole mRNA will be consisting of introns and exons, but basically in the mature mRNA when it's basically about to get translated to a protein. In case of eukaryotes, the introns are cut off and the exons are kind of ligated together to form just the exons of the mRNA, which gets translated downstream. Just a second. तन्मय तन्मय वो कृतिका दी टेक्स्ट कर रही है कि वो एक्स क्रोमोसोम के बारे में बात कर रहा है अटेंड करने के लिए थोड़ा अटेंड कर लेना तो बताना मेरे को. Yeah, so in eukaryotes, you have this mechanism of like splicing where your only the exons are allowed to be translated into amino acids, the introns are cut off. So, uh, so this is a major issue in terms of read alignment, but we'll see in the later stages, because when you're aligning to the genome, this will not be aligning in like one of the reads won't be overlapping in the intron regions. Only the exons will have the reads because the introns are not present in the mature mRNA. So that is an issue that we'll see in the next slides, but that is an issue that you need to keep in mind while kind of mapping RNA-seq reads. So com- over 90% of human genes are alternatively spliced to form two or more distinct expressed isoforms. So these are known as RNA isoforms. So a particular mRNA can be spliced in different manner. So if you have, say, like in say for example, I'm giving just an example. You have a read. You have like alternative exons and introns for 6 times. So first comes intron 1, then exon 1, then again intron 2, exon 2, like this. Now it can happen after splicing. It's just the exons are kind of ligated in the same order as it was there in the genome. So the read will look like exon 1, exon 2, exon 3, and it will go on till exon 6. Now it can also happen कि exon 3 comes first, then comes exon 2, then comes exon 4, like this. So there can be different types of these combinations of these exons which can be made into the mature mRNA. So these are like called as different isoforms of the same mRNA. So it's actually the same, it's coming from the same portion of the genome, but the mRNA is spliced differently. So the downstream proteins that will be formed will be kind of different compared to its isoforms. So the complexity of the information captured increases from short read cDNA sequencing, sorry, where isoform detection can be compromised by reads that cannot be mapped unambiguously to long read methods that directly sequence isoforms. In short-read cDNA sequencing, a significant portion of the reads map ambiguously when an exon is shared between isoforms. Reads that span exon-exon junctions can be used to improve this isoform analysis, but also can be mapped ambiguously when a junction is shared between isoforms. These issues complicate analysis and the interpretation of results because it's quite evident because there can be different isoforms of the same gene, but you need to correctly identify which isoform this is and align it uh, in that manner. Okay. Long-read cDNA methods, on the other hand, can generate these full-length isoform reads that remove or substantially reduce these artifacts to improve differential isoform expression analysis. However, these methods rely on cDNA conversion, which removes information about RNA-based modification. This is what I was saying. You cannot directly sequence the RNA, so you're converting it back to a DNA, but you are losing out information because of that, which includes polyadenylation. You don't know if polyadenylation was there or not because you're anyway removing it because you cannot uh, sequence that. So polyadenylation presence or not, or if there has been other kind of modification on the mRNA, base modification detection such as N6 methyladenosine and poly-A tail length extension. So when you're converting the RNA to the DNA and sequencing it, you are losing a bit of information in the context of the RNA, just that you do not know what kind of modifications, what is there, what kind of other things were there. So slight loss of information in this method. So this is another method which basically is the single-stranded RNA or non-stranded or unstranded RNA sequencing. So the stranded protocol differs from the non-stranded protocol in two ways. First, during cDNA synthesis, the second strand synthesis continues as normal, except the nucleotide mix includes DUTPs instead of DDTPs. So like it's evident because in RNA in place of thymine you have uracil. So in basically during cDNA synthesis, the second strand synthesis continues as normal, except the nucleotide mix includes DUTPs in case of DDTPs. And soon after library preparation, a second strand digestion step is added. This step ensures that only the first strand survives the subsequent PCR amplification step and hence the strand information of the libraries. So basically you have this mRNA fragment and in non-stranded case you incorporate DDTP, DTTP, sorry, DTTP, not DDTP, DTTP. So deoxy, uh, basically thymine phosphatase. The DTTP. So in case of non-stranded, you use the thymine version of it. In case of stranded, use a uracil version of it. And when it is broken off prior to PCR amplification, the DUTP mark strand is selectively degraded by this uracil DNA glycolysase enzyme. So the kind of if there is a uracil present, it will selectively degrade that strand, and the remaining strand is amplified to generate a cDNA library suitable for sequencing. Now we kind of went through the different types of RNA sequencing protocols and what is important and like how do we do that basically and kind of what are the different challenges that we face, long read and short read. Now comes the alignment part of the yeah. Yeah, now comes the alignment part of the pipeline. So the alignment will be slightly different because as you can understand, uh, in DNA, you are kind of aligning the whole DNA or just so the alignment tools that are used for DNA sequencing is kind of different from the alignment tools that are used for RNA sequencing. So one of the most popular RNA sequencing methods is called the top hat, which aligns in two steps. So these are like splice aware aligners. So unspliced reads are mapped to locate the exons, and the unmapped reads are then split and aligned independently to identify the exon junctions. The RNA sequencing alignment program currently is used by the Expression Atlas pipeline, which is basically the EMBL European Molecular Biology Laboratory pipeline. Uh, is called HiSat2. This is another software that is in comparison with Top Hat. It stands for Hierarchical Indexing of Spliced Alignments of Transcript. And it's the second version. And it provides more accurate result with fast and sensitive alignment. Uh, HiSat2 uses graph-based alignment approach to index the reference genome combined with the bow tie 2 algorithm for alignment, which is the BWT and everything you do. So it is important to check the quality of the mapping process. The percentage of mapped reads is a global indicator of the overall sequencing accuracy and of the presence of contaminating DNA. And here you also can use Picard for quality controlling mapping. So we'll look into some of these alignment tools and how these work in the subsequent slides. So one of the most popular alignment algorithms or softwares you can say is the star alignment. So this is the website. And this is basically the GitHub, and like you can install it from here, and this is the paper for star aligner. So this is a very widely used and very commonly used uh, software that for RNA sequencing alignment if you want to have a better read. Anyway, let's continue. So this what it does is basically there are a couple of things. The first thing is seeding, and the next thing is clustering, stitching, and scoring. So we'll go through this one by one. So star is shown to have high accuracy, and it outperforms other aligners by more than a factor of 15 mapping speed, but it is memory intensive. The algorithm achieves this highly efficient mapping by performing these two steps: what is seed searching, and the second step is clustering, stitching, and scoring. Now what is seed searching? For every read that this algorithm aligns, this star will search for the longest sequence that exactly matches one or more locations in the genome. So it's very simple. You have the read. You're searching for the longest sequences that exactly matches one or more location in the reference. It's good enough. Now these longest matching sequences are known as MMPs, which are known as maximum mappable prefixes. So from the name also it is quite understandable. It's the longest substring that can be found inside the genome. Now, these different parts of the reads that are mapped separately are called seeds. So the first MMP that is mapped to the genome is called seed 1, and like it can map to different portion of the genome, right? So one, like if it matches to one portion it is seed 1, if it matches to another portion it is seed 2. So like there are like why is it matching to different portions in the genome? So the simple answer is because of splicing. So in case of eukaryotic genome, what we'll have is the introns will be cut off. So the portion, so if this is a reference genome, the whole read won't be aligning to the same, like the whole area in the reference genome, because this part did not come from here. This part came from another exome, which is like say for example 200 kb upstream of this of that particular area in the genome. So like it'll read the read will map in two different places. So the different parts of the read that are mapped separately are called seeds, and the first MMP that is mapped to the genome is called seed 1. Then it will search again for the only unmapped portion of the read to find the next longest sequence that exactly matches the reference genome on the next MMP, which is known as seed 2. So you have this is the whole read. One portion of the read matches here, the another portion of the read matches here. So this is seed 1, this is seed 2. The sequential searching of unmapped portion of reads underlies the efficacy of the star algorithm. It uses an uncompressed suffix array I hope you remember what suffix arrays are, to efficiently search for MMPs, and this allows for quick searching against even the largest reference genomes. Other slower aligners use algorithms that often search the entire read sequence before splitting the reads and performing iterative rounds of mapping. So it, it is better in that way. Like it does not try to align the whole seed altogether. First only it splits it up and tries to align the maximum substring that it can align, and then it tries to align the remaining part. No, now, like what does this actually mean? So seed one is aligning here, seed two is aligning here. So this portion has been spliced off. So this is an intron that has been spliced off, and this is the donor side, this is the acceptor side, right? So and this portion of the genome has been spliced off most probably because it was an intron. Now the sequential if star does not find an exact matching sequence for each part of the read due to mismatches or indels, the previous MMPs will be extended. So say for example, you there's a mismatch here, or there is some deletion or some sort of mismatch here. What it'll try to do is it'll try to extend this on to the left. It'll try to kind of try to extend the previously mapped regions and try to see if that portion is matching to this portion. If the extension, if after even doing the extension, if it does not give a good alignment, that then the poor quality, the adapter sequence or other contaminating sequence will be soft clipped. So this read will be some clipped saying, okay, like this is a bad quality read, or there has been some contamination, or that adapter sequence is still there. So in those cases, it will soft clip those parts and align the rest of the read to the genome. Now, this technique where it considers that like a read can be originating from separate portions of the genome. So these are what are known as splice-aware aligners. So basically, it takes into account the splicing of the RNA and it tries to align the reads in a similar fashion. It does not try to align the whole read into one place because most probably it has not generated from the one from a single portion of the genome, because most probably the DNA RNA has been spliced before expression. Now, once you have done this, like found where the reads have been originated from, the separate seeds are then stitched together to create a complete read by first clustering the seeds together based on proximity to a set of anchor seeds or seeds that are not multi-mapping. Then the seeds are stitched together based on the best alignment for the read, scoring based on mismatches, indels, gaps, etc. So, like after that has been done, it kind of like extends. This is a single read, but this is a stitched read which has been kind of separated. So this portion aligning to this portion aligning to this portion. It's a single read, but it is now in the form of a stitched read. And also there has scoring techniques involved here, where it will like kind of uh, uh, penalize for mismatches or indels or different types of other variations. Okay, so this is a kind of a like a table containing all the major read mapping algorithms that we have. So if you, for botai2 also has a transcriptomic alignment reference, which is can directly align it with the transcriptome. But in most of the cases, they either use the genome or the genome and the transcriptome as well. So STAR is one of them. TopHat and HiSat2 are the next like kind of most popular algorithms out there, but STAR is kind of the most widely used. Okay, now this was like read mapping in terms of transcriptomics. Now, another method would be to assemble the whole transcriptome. So you have raw reads, and then you process these reads by removing the adapters and other type of contaminations. And what you try to do is a de novo assembly, and you do some quality control on that. And you do like after read mapping, you find like how many reads are mapping to where, and like how many reads are originating from this gene, how many reads are originating from that gene. That tells you how much a gene is getting expressed in that particular environment. So you'll do statistical testing on this. Basically, condition A, you'll have a particular gene having more transcripts than other particular gene, and if you like compare these different conditions, what you will get is basically an idea about in which condition a particular gene is expressed higher, in which condition the particular gene is like expressed lower, and this type. A transcriptome constructed from short-read RNA sequencing is an easily attainable proxy catalog of protein coding genes when the genome assembly is unnecessary, expensive, or difficult. So what will happen is in many cases, what you do if you want to check expression of particular genes in organisms that do not have a very good complete reference genome. So in those cases, what will happen is it's a tedious process to do that because you don't have the reference genome, and also like there are other problems, and it can be like computationally expensive and difficult as well. So in those cases, what you'll do is basically try a de novo assembly technique. So in the absence of a sequence genome to guide the reconstruction process, the transcriptome must be assembled de novo, only using the information available in the RNA sequencing reads. Subsequently, the sequences must be annotated in order to identify sequence intrinsic and evolutionary features in them, for example, protein coding regions. Although straightforward at first glance, de novo transcriptome assembly and annotation can quickly prove to be challenging undertakings because obviously the amount of information that you have is very low. Okay, so this is a graph-based approach to de novo transcriptome assembly. The basic idea is to establish a catalog of substrings from the RNA-seq reads and compose these into a graph or a set of graphs, wherein the substrings are connected if an overlap between them exists. This establishes path to graphs which correspond to the reads or potentially originated from. A short nucleotide reads of 50 to 200 nucleotides in length are the inputs for the assembly process. If the paired end reads are supplied, the respective mates are merged into a single contiguous read prior to assembly. Highlighted here is a six nucleotide position of a single read. This is the read. These are subdivided into subsequences of definite length k-mers. So for each read, all possible subsequences of length k are generated, which are known as k-mers. In this case, they are like k equal to 4 here. They are they kind of divided into reads of length equal to 4. So these are known as 4 mers. The 4 mers originating from the 6 nucleotide fragment from the previous step are indicated here. So these are the 4 mers. These are the all possible formats that you can get. Now, how do you kind of assemble these reads? Subsequently, each k-mer becomes a node. So in terms of this is done usually using graph theory. So basically, if you imagine a graph, your nodes are basically your reads, which are your k-mers, and these nodes are connected by edges uh, if they satisfy overlap between these two reads. So, for example, in one node you'll have GTTA, another node you'll have TTAG. These two nodes will be connected because TTA is common. So the edge will represent the common substring between these two reads, these two k-mers, if I may say, and and basically they'll be connected only if there is a overlap between these two reads. The edges are established between any two nodes that satisfy this overlap requirement. So in this case, the requirement is at least k minus 1 nucleotide overlap with each other. So in this case, like 4 reads, if k mod is k equal to 4, then should be at least 3 overlaps between two reads to be connected. So yeah, and yeah, and subsequently uh, uh, what happens is you build out this whole graph, and this graph will consist of nodes which are reads, and the edges will be connecting them if there are overlaps between these reads, and what you form is basically a De Bruijn graph. We'll talk about De Bruijn graphs in genome assembly in the later weeks. I think in week 9 or 10 there is this part there. We'll talk in much more greater detail in that class. But this is the graph that you are forming is basically a De Bruijn graph. Now, if you want to find the contiguous sequences, you need to do an Eulerian walk over the graph. So basically, move from one node to another to another to another to get the whole sequence like that. So like technique of building De Bruijn graphs and then doing an Eulerian walk to find the sequence of the whole sequence is basically a similar technique is used for genome assembly as well. So this is de novo transcriptome assembly, but in case of de novo genome assembly as well, we use a similar technique where you have these nodes and form the whole graph, and you want to find what is the longest connected component of this graph, and you kind of do a Eulerian walk on the graph, right? Finally, different parts of these graphs are traversed and recovered as independent sequences. Not all paths to the graphs are recovered. The subset of paths that represent valid transcripts is determined algorithmically. Now, uh, now we come to the concept of transcript quantification. So, first of all, why do we need to quantify transcripts? This is the first question that arises. Estimating transcript abundance is a fundamental task in genomics because, excuse me, these estimates can be used for classification of disease and their subtypes, for understanding expression changes during development, and also tracking progression of different diseases like cancer. Accurate and efficient quantification of transcript abundance from RNA-seq data is an especially pressing problem due to both the wide range of technical biases that affect the RNA-seq fragmentation, amplification, and sequencing process, the exponentially increasing number of experiments, and the adoption of expression data for medical diagnosis. In case of RNA-seq, the features are typically genes. So basically, what will happen is in your file, if you have a like gene expression matrix file on your row, like basically the first column will be your genes, and the the other columns basically will tell you the different samples, and each of those elements will tell you the expression of that particular gene in that particular sample. So these features are typically genes where each gene is considered here as a union of all its exons. One may also consider each exon as a feature. That becomes more complicated. Example, in order to check for alternative splicing. For comparative chip-seq, the features might be a binding region from a predetermined list. Special care must be taken to decide how to deal with reads that overlap with more than one feature. Uh, HTSeq count is a software that helps us that allows us to choose between three modes of three possible conditions from this. Uh, if none of them fit your needs, you can obviously write your own script with HTSeq. The three overlap resolution modes of HTSeq count work as follows. For each position I in the read, a set Si is defined as a set of all features overlapping that position. In this case, genes, all the genes overlapping that particular position. Then consider the set S, which is with I running through the position we need. S as the union of the sets of Si for mode union, the intersection of all the sets of Si for mode intersection stripped, and the intersection of all non-empty sets Si for mode intersection non-empty. These are the three modes that are available for HTC counts. So if S contains precisely one feature, the read is counted for this feature. If it contains more than one feature, the read is counted as ambiguous, not counted for any features. And if S is empty, the read is counted as no feature. This is the figure that kind of explains these different conditions. So if a read is here, say for example, in the first case, if the read is only covering gene A, so the union intersection and the intersection on empty all will be like gene A. But if we take a if we take a different example, like for this one, so this read kind of spans two genes, right? So in case of union, you'll have basically this is This is the same gene but different exons. So in case of union, you'll have gene A. This read will be transcript will be quantified for gene A. If there is a if you are using the intersection strict, there will be no feature, because in intersection strict, it's the intersection of all the SI sets. It's not the non-empty sets. It's all the SI sets. So there will be no feature. And intersection non-empty, there will be gene A. Now let's take a more complicated example like this one. So this read is mapping to two regions. So union will be ambiguous, intersection will be ambiguous, and intersection non-empty will be ambiguous because it is directly overlapping the common area between gene A and gene B, right? Now this is a more interesting example. So this read aligns fully to gene A, not fully to gene B, but also aligns to the middle portion of it. So if you take the union, this is ambiguous because it is kind of aligning everywhere. If you take intersection strict, it is like fully aligning to gene A. To gene B, it is aligning only partly, so it will be gene A. And if you take non-empty intersection as well, it will only align to gene because it is aligning wholly to gene, right? So this is how quantification of different transcripts are done. So this is a standard kind of method that HTC count follows. You can use your own kind of logic for this. Yeah. So yeah, and so these are the different types of conditions in HTC counts. But if you want to like have your own logic, you can just modify the script of HTC count. But generally, like this is the method that we follow. So if it is actually kind of overlapping one particular gene fully, another gene like not like fully, partially, we generally put it in ambiguous, and we do not count this transcript as a read for any of the features, in our case the different genes, gene A and gene B. So this is the last uh, portion of the slides. So we talked about HTSIC as HTSIC counts as a software that can be used for transcript quantification. RSIM is another software that kind of does this transcript quantification thing. So RSIM, the full form is RNA-seq by expectation maximization. Expectation maximization, on the other hand, is basically an algorithm which kind of iteratively checks and tries to minimize or maximize something, right? So it's a iterative process, and like it'll have a cost function which will try to minimize or maximize. So RSEM, which implements our quantification methods, provides extensions to our original model. The key feature unique to RSEM is the lack of the requirement of a reference genome. So this is very important. So RSEM does not require a reference genome, so that is why it is also like it uses this method for iteratively checking. So that's why it does not require a reference genome. Instead, only it requires the user to provide a set of reference transcript sequences, such as the one produced by a de novo transcriptome assembler. After alignment of the reads, excuse me, RSIM computes the maximum likelihood abundance estimates using the EM algorithm for its statistical model. A number of options are available to specify the model that is being used by RSIM, which should be customized according to the RNA-seq protocol that is produced that produce the input reads. For example, if a strand-specific uh, protocol is used, you use the strand-specific command. There can be other things as well. The fragment length of the distribution can be controlled as well. For PE analysis, RSIM learns the fragment length distribution from the data. If the protocol produces read position distributions that are highly 3' or 5' based, then the estimate RPST option should be specified so that RSIM can estimate a read start position distribution, which may allow for more abundance estimates. So this is the RSIM paper. If you want to read it, it like kind of very well explains it. What are the different types of algorithms and how it checks it. But yeah, the thing to remember from this particular algorithm here for our cases is basically it uses expectation maximization. That's all. Okay. Okay. This brings us to the end of the discussions. Now we'll move into the assignment part. Sorry. Write the answers in the chat if you want. Okay. So for copy number variants, these are structural variants actually. That these either increase the amount of specific DNA in the genome, or it decreases it. So for duplications, the copy number will increase. For deletions, the copy number will decrease. So the first option is correct. CNV occurs when there are changes in the order of the DNA segments in the genome. That is not true. So if there are changes of order in the genome, so for example, orientation in the genome, a particular area of the genome, the orientation of the reads has been changed, so that indicates an inversion. So, during an inversion, there is no change in the amount of total DNA that is actually present. So, the inversion is not a CNV; it's a non-CNV. Right. So option B is wrong. CNV can occur both at non-coding and coding regions of the genome, which is actually true. There is no limitation to that. The CNV has to occur in this particular area of the genome, so there is nothing like that. And deviations in the read depth from the expected copy number can indicate the presence of CNVs. And actually, this is how CNVs are detected. So if your general, say for example, coverage is 30, in the place of the CNB, if there's a duplication, the coverage will suddenly increase to 100 or 120, like that, or it will suddenly go down to 5. So for a deletion, so this drastic changes in read depth and this deviation in read depth from the expected copy numbers, these are actually indicative of the presence of a CNB. So options A, C, and D are correct. Moving on to the next question. So the first option, insertion, inversion, and translocation are different types of non-CNV structural variants because uh, the copy number, the amount of DNA does not change. So inversion and translocation just uh, like translocation is like inserting a sequence from somewhere else. Inversion is just uh, like uh, changing the orientation of the reads and so on. So these are not copy number variants. So these are non-copy number variant structural variants, non-CNVs. And CNVs are not part of structural variants. That is not true. CNVs are actually a part of structural variants. It's a specialized part of structural variants. Non-CNV SVs can lead to formation of multi-allelic CNVs, which is not true. So there is no connection between having multi-allelic CNVs to like with non-CNV non-CNV SVs, because like with like if the SV is not CNV in the first place, there will be no formation of multi-allele. Okay, so yeah, and non-CNV structural variations involve changes in the structure of the DNA, but they do not involve changes in copy number. That is true. The changes in copy number will only be happening for CNVs. For non-CNVs, the structural variants will be there, but they won't change the copy number. So, okay, moving on to the third one. Okay, so if you see this, this is the difference, like all the reads are oriented in a similar fashion. In the sample, the orientations are a little bit off. So this is basically a paired end duplication inversion. So first of all, so this is ABCD. Now, if you look into it, D has been duplicated twice, A has been duplicated twice, B is not been duplicated, and C has not been duplicated. So on the end portions between A and D, like in the end portions, the reads has been inverted and duplicated. So D has been inverted there and duplicated, and A has been inverted here and duplicated. So basically, it's a paired duplication inversion. So it's a complex structural variant. This is not a normal structural variant. This is a complex SV. But at the end of the SV, the read pairs have been duplicated and inverted. So it's a paired duplication inversion. Okay. So the question is, which of the tools use read pair distance method for identification of structural variant? The answers are breakdancer and PEMR. So if you go back to this slide, so breakdancer will be somewhere here. Yeah. So it uses art read pair, and I'm not sure if PEMR is here. No, it's not here. I'll just let's go back here. So this is the paper for PEMA. You can go through it, but it uses. read. Uh, it's not written here, but if you go through the paper in detail, it will show you that it also uses read pair technique to identify SVs. I'm not going into the details because I have not discussed this particular software, but yeah, if you want, you can just go through the paper once. Okay, the question was basically what is the kind of evidence you will be looking for detecting small SVs whose breakpoints are located within repetitive or complex genomic regions. So split read uh, and assembly-based methods are the answer. Why not read pair and read depth? Because read depth, if you kind of think of it, the depth is determined by how many like reads are supporting one base pair. But in if it's a genomic repeat region, what will happen is the reads will not align very like properly because in repeat region the regions are very similar. So a read can go and map to a different repeat than what originated from. So read depth methods are not very useful in terms of complex genomic, as in repetitive genomic regions, which are complex in nature. Now, read pair distance method is the same thing. If there are opposing pairs of reads that are aligning, and you are estimating the distance between them, and then you are trying to say if there is a SV or not. So that also won't work very well because there are repeats around the region. So the lead pairs that are going to align to the genome, they can be discordantly aligned anyway because of the repeats. So that does not mean that there has been a SV. So split read is something that is very good in determining these kind of complex SV, complex region SVs, because like split read is something of a definitive proof that there has been a structural variant, and obviously assembly based methods where you do de novo assembly and check. So that is always there, but split readism is a better evidence to have than read pair or read depth methods. Moving on. Okay, I'll not dwell in this question much because I've not discussed this, but yeah, the, for feature counts algorithm, you need SAM GFF or BAM GFF, SAM or BAM, anything will work. And also in place of GFF, you can also have a VSIF file. So I'll just quickly go to the feature counts. So this is a demonstration that shows feature counts. Sorry, it takes a GTF annotation file, or you can also give a VCF annotation file. It takes a alignment BAM file, or you can also get the alignment SAM file as well. So yeah, moving on. Okay, so the answer is C. I'll just go over it again. Alternative splicing is basically you have a set of like sequence of exons introns inside the mRNA, but not necessarily the exons will be joined in the same order that they are present in the genome. So exon 1, 2, 3, the order might not be the same in the RNA, and that can be like exon 3 to 1 like that as well. So these are what are known as RNA isoforms because they are originating from the same DNA sequence, but due to their splicing difference, the RNA that is formed downstream and the protein that is formed like after that, it's not basically the same. So that's why they are like different, but they are originating from the same DNA sequence. So these are known as mRNA isoforms, and these generate these are generated due to alternative splice. Just, sorry. Okay, the answers are A and D. That the advantages of long read direct RNA sequencing is it can detect RNA modifications which you cannot detect once you convert the RNA back to a cDNA. So that's one thing for the direct RNA sequencing. And if it's a long read, it's also been good, also be good in detecting isoforms. So yeah, these two options. Moving on. The question is which of the sequencing processes is useful for analyzing expressions of two genes that overlap with each other on opposing strands? Small RNA sequencing. I have not touched on the topic, so I'll just quickly. So, in Illumina's website, you can see it's basically the small non-coding RNAs act in genes like silencing and post-transcriptional regulation of gene expression. Small RNA sequencing is a technique to isolate and sequence small RNA species such as microRNAs or snRNAs or shRNAs. There are different types of small RNAs. The small RNA-seq query can small RNA-seq can query thousands of small RNA and miRNA sequence with unprecedented sensitivity. With small RNA sequencing, you can discover novel miRNAs and other small non-coding RNAs and expression uh, and differential expression of all small RNAs in any sample. Okay, so if there are like two genes that overlap with each other on the opposing strand, the best way to do it is basically small RNA sequencing. Okay, this is a fairly straightforward one. So ISA2 uses a combination of FM and Burroughs-Wheeler transform, FM indexing and Burroughs-Wheeler transform to construct these indexes. It's kind of very similar to BOTI2, and it's also a splice-aware alignment. Uh, these are not true. It only uses BWT, and like these first two options are false. And it also includes a module for SMP calling as well. So that's not true as well. Okay. Okay. Uh, Star follows sequential seed searching as we saw from the slides as well. So you have a read searches for one portion of the read, the seed one, second portion, seed two. So it does that sequentially. And reads are matched for maximum mappable prefix against a reference genome sequence. But this is false. The algorithm divides all reads into small fragments. What it does, it basically try the maximum matching for to the sequence. It does not divide the read at first. After it has been both the sides of the read has been mapped to different parts, then it attaches a string between them to depict like they're coming from two different parts of the genome, but it does not essentially split the reads. And seed searching requires building suffix trees from the reference genome. It's actually they use suffix arrays, not suffix trees. So The answer is except HiSec 2, all of them use the alignment free method, and none of them we discussed. But Salmon is a very popular one. You can take a look. I'll just send you. There's a tutorial in GitHub for Salmon. So this is a very good tutorial for Salmon if you want to go through. Uh, Okay, we read about BWT in the last to last class, I think, if I can remember well. Yeah. So yeah, it is used to compress reference genomes. It is used to compress any kind of a string or any kind of file. BWT is used for that. Uh, it also is used for alignment, for memory efficient alignment of sequencing reads. We saw that virus Wheeler transform, BWA, BM, those stuff. Uh, it is actually not responsible for handling alternative splicing events in the reference. BWT does not have any functionality in that, but it is used to find exact matches between weeks and reference as we saw in the alignment stage. So yeah, only C is incorrect. This is the last question. Okay, the answer is RSM that we already discussed. Another is Callisto. So this is another tool that uses this EM algorithm for isoform quantification. Okay, so that's all for today. If you have any questions, you might ask, or we'll stop this session. Okay, then, if there are no questions, thank you for joining. We'll meet again next week. Thank you.